Assalamualaikum, sir. This is Dr. Maham. I have a question for you. What makes methicillin-resistant Staphylococcus aureus a superbug?
Wow, that's a great question. Dr. Maham is asking what makes methicillin-resistant Staphylococcus aureus a superbug. I mean, why methicillin-resistant Staphylococcus aureus is so difficult to treat? Why so much mortality and morbidity is associated with the infections of MRSA? Now, in the lecture now onward, rather calling it full name, methicillin-resistant Staphylococcus aureus, I will be calling it MRSA. That is very commonly used in medical community, but casually we can also call it MRSA. Now let me first define what is MRSA, what is MRSA, what is methicillin-resistant Staphylococcus aureus, right? This is a very special type of Staphylococcus aureus which has a unique gene, right? It's a very special type of Staphylococcus aureus strain which has a very special gene, right? Now, what is that gene? That gene is called Mech A. Mech A means MEC, A for apple. Mech A. When any Staphylococcus aureus, whenever any Staphylococcus aureus acquires Mech A gene, it becomes MRSA. Now we can say another way. Number one, I said, I will explain the mechanisms later. First thing, listen carefully, I'm repeating. Any Staphylococcus aureus which acquires Mech A gene or Mech C gene or has Mech A gene or Mech C gene, Mech C, C for cat, A for apple. Such Staphylococcus aureus is called MRSA. What does it mean? It means that if any Staphylococcus aureus is genomically altered in such a way that it has Mech A or Mech C gene, right, it will become resistant to methicillin. That was the original concept, right? That was the original concept. But the real thing is that when we say that a given Staphylococcus aureus is methicillin-resistant Staphylococcus aureus, of course it is understood that this Staphylococcus aureus is carrying Mech gene, and of course it is understood that this should be methicillin resistant. But this is a little part of the story. You know what is the real story? Such Staphylococcus aureus, which has a Mech gene, not only it is resistant to methicillin, but it is resistant to almost all beta-lactam antibiotics. Listen with your both ears. That when I say that patient has MRSA or patient has methicillin-resistant Staphylococcus aureus, it does not mean that patient has a Staphylococcus aureus which is only resistant to methicillin. It is misnomer. It's a wrong name. You know what should be the real name of such Staphylococcus aureus? The real name should be almost all beta-lactam resistant Staphylococcus aureus. What should be the real name? Almost all beta-lactam antibiotic resistant Staphylococcus aureus. What I mean by that? I mean by that Staphylococcus aureus, which has Mech A gene or C gene and expressing its products, it becomes resistant to all major classes of beta-lactam antibiotic. It means it is resistant to penicillins, it is resistant to cephalosporins, it is resistant to monobactams, it is resistant to carbapenems. Even within the group of carbapenem, you must be knowing meropenem is broadest spectrum antibiotic, one of the broadest spectrum antibiotic. MRSA is even resistant to that. Now, how it happens? what really goes on at molecular level and genetic level. We'll start step by step, right? First, I will talk about that when Mech A gene and is present in a Staphylococcus aureus, what changes come into Staphylococcus aureus, right? Number one, Staphylococcus aureus has acquired Mech gene, Mech A or Mech C gene. Now, this gene, when it expresses itself, right, it makes a special proteins, right, which are called penicillin binding protein 2A. Now, penicillin binding proteins we are going to talk about? Yes. Actually, all staphylococci have different types of penicillin binding protein. On the membrane of Staphylococcus aureus, normally there is penicillin binding protein 1, penicillin binding protein 2, penicillin binding protein 3, penicillin binding protein 4. This is normally present on Staphylococcus aureus and many other bacteria. Of course, many other bacteria, but right now we are focusing on Staphylococcus aureus. So, what really happens that those staphylococci which are not methicillin resistant, right, they are having multiple penicillin binding protein 1, protein 2, protein 3, protein 4. But what is very special about methicillin resistant Staphylococcus aureus, or what is special about MRSA, or what is special about MRSA? At genotypic level, at genomic level, it has a gene, additional gene, which is called Mech A. I will not every time repeat Mech A or Mech C. Let's suppose we just use Mech A. So, at genomic level, those Staphylococcus aureus strains which have acquired Mech A gene, right, they express altered penicillin binding protein. They expressed altered penicillin binding protein, and that altered penicillin binding protein, which is being expressed on such staphylococci, are called penicillin binding protein 2A. Should I tell you? This is also misnomer because it does not bind penicillins, but because this altered Mech A makes mRNA, and which translates into eventually a protein which is somewhat modified penicillin binding protein 2. So we call it penicillin binding protein 2A. Now, this penicillin binding protein 2A, it is only present on those Staphylococcus aureus which are containing Mech A gene, right? So what happens? That whenever a Staphylococcus aureus acquires Mech A gene and expresses it, then its surface membrane expresses penicillin binding protein 2A. Now, of course, normally Staphylococcus aureus are having penicillin binding protein 1, 2, 3, 4, but not 2A. They have penicillin binding protein 2A only when Mech A gene is present. Of course, these protein, penicillin binding protein 1, 2, 3, and 4, they are present from thousands of the years, maybe millions of the years. I don't know. I was not there. But why we call them penicillin binding protein? Because penicillin was discovered last century. But bacteria were having these proteins from long time. So what is the normal function of this protein? We have to see what is the normal function of penicillin binding protein. Of course, penicillin binding proteins were not produced by the nature to bind the penicillin, right? They have some other function. So let me explain first that function. right? The function is, what is the function of penicillin binding protein? Let me explain. Penicillin binding proteins are normally expressed on the cell membrane of the growing Staphylococcus aureus or even grown Staphylococcus aureus. Now, what is their function? Let me explain. Actually, when Staphylococcus aureus is growing, it synthesizes, and secretes around it peptidoglycan chains. When Staphylococcus aureus is growing, it synthesizes and secretes peptidoglycan chains. Now, when these peptidoglycan chains go out of the Staphylococcus aureus and they are accumulating around the Staphylococcus aureus, at that very moment, penicillin binding proteins actually cross-link these peptidoglycan chains. So what is the normal function of penicillin binding protein? Penicillin binding protein basically cross-link the peptidoglycan chain which are synthesized and secreted by the Staphylococcus aureus. And when these chains are cross-linked, they make a very strong meshwork or network around the bacteria. And this cross-linked meshwork of peptidoglycan around the bacteria is called bacterial cell wall, right? So what penicillin binding proteins are doing? We can simply say the basic function of penicillin binding protein is that they should cross-link the peptidoglycan chains so that a strong, healthy cell wall can be made and maintained around the bacteria. Here, Staphylococcus aureus. Now, when initially penicillin was discovered, let me go in the history a little bit. I will come back to MRSA. Let's go to the history first. When penicillin was newly discovered and then we started using it in 1940s. 1940s, when we started using penicillins as antibiotics, especially natural penicillins like penicillin G and penicillin V, what happened? What these penicillins were doing? How they were killing the bacteria? Let me explain. Actually, penicillin binding protein were binding with these enzymes which were doing the cross-linking of cell wall components. Because these penicillin binding proteins are naturally there to cross-link the peptidoglycan, they are functionally named as transpeptidases. I will repeat it. Up to now I was telling that bacteria on their membrane, they are having penicillin binding protein. But original function of penicillin binding protein is cross-link the secreted peptidoglycans to strengthen the bacterial cell wall, right? This function is transpeptidation. So we can say penicillin binding protein has basic function of transpeptide function. They function as a transpeptidase, or they have an enzymatic function of transpeptidase, which is responsible to cross-link the glycoprotein chains to strengthen the cell wall of the bacteria, right? Now, in 1940s, when we started using the penicillins, penicillin have beta-lactam ring, right? Penicillin has two rings, but I'm specially focusing on beta-lactam ring. Now, penicillin's beta-lactam ring will bind with the penicillin binding protein. Or, of course, we can say penicillin beta-lactam ring will bind with the transpeptidases. Now imagine a bacteria which is trying to grow. It is a small bacteria and it is trying to become bigger, and it is secreting peptidoglycans. But unfortunately for the bacteria, we have released the penicillins on this bacteria. Our penicillins will bind with penicillin binding protein, and penicillin binding protein, when they are bound with the beta-lactam ring, they stop working. They stop working. So what is the purpose of beta-lactam antibiotics generally speaking? The purpose of beta-lactam antibiotics, which include penicillin, cephalosporins,
carbapenems,
monobactams, and carbapenems. What is the purpose of penicillin, cephalosporins, monobactams, carbapenems? Their main purpose is that these antibiotics do have beta-lactam ring, and they should go and bind with penicillin binding protein and make them dysfunctional. Now you imagine whenever beta-lactam antibiotics or penicillins are given to the bacteria and transpeptidase bind with the penicillin binding protein, transpeptidases become dysfunctional or non-functional or less functional. What will be the result? Result will be this, that bacteria will still keep on secreting peptidoglycans. But because transpeptidases or penicillin binding proteins, they are dysfunctional, so they will not cross-link the peptidoglycans. And when peptidoglycans, which are the components of the bacterial cell wall, if they are not cross-linked, they don't make a proper tight network or meshwork, what really happens? Bacterial cell wall become very weak. Now, if bacterial cell wall is very weak, all of us know that interior of bacteria is usually hyperosmotic as compared to the surrounding environment. So, what will happen? A bacteria with normal cell wall, even if it is hyperosmotic, it will absorb little water, it swell little, but if cell membrane is well cross-linked and healthy, it will not allow the bacteria to burst. But when bacteria are exposed to beta-lactam antibiotics, which will bind with the penicillin binding proteins, and penicillin binding proteins fail to do the transpeptidation, and protein and glycopeptide chains are not cross-linked, and cell wall is weak, then bacteria will start absorbing the water and it will keep on swelling until it bursts. So what penicillins are doing? If penicillins are really functioning on target bacteria, they will lead to osmotic burst of this bacteria. That is why beta-lactam antibiotics are called bactericidal. They kill the bacteria, right? Now we come back to that in 1940s medical community was very happy that we have natural penicillin, that we are bombarding these penicillins on the bacteria, and bacteria's cell membranes are not cross-linked and we are able to kill the bacteria, right? But then bacteria got clever. It's a little history. You know what happened? Bacteria started producing a special type of enzyme. Bacteria started secreting a special type of enzyme which could destroy the beta-lactam ring. What happened? We were using natural penicillins. Natural penicillins were binding with the penicillin binding protein, the transpeptidases. Failure of transpeptidases will leading to failure of cross linkage of cell membrane components and leading to bacterial death. But then some resistant bacteria came. Then doctors investigated, there are other bacteria, we are putting penicillin G or penicillin V or some natural penicillin on them, but they are living happily, they don't mind. What is happening there? Then we discovered they are very clever bacteria. They have acquired some genomic component and they have undergone certain mutations and they have started producing a special type of enzyme, and that enzyme was secreted by the bacteria, and these enzyme which was secreted by gram-positive bacteria into extracellular environment and by gram-negative bacteria in periplasmic space, these enzymes which were secreted by the bacteria, they had the capability to destroy the beta-lactam ring. right? So we named these enzymes beta-lactamases. Beta-lactamases. My very good friends, don't confuse beta-lactam antibiotics with beta-lactamases. Beta-lactam antibiotic is an antibiotic which is having beta-lactam ring, like penicillins, cephalosporins, monobactam, carbapenem group. But beta-lactamases are not antibiotics. Beta-lactamases are enzymes which are produced by the bacteria in their defense. And what beta-lactamases are doing, they bind with our beta-lactam antibiotic and destroy the beta-lactam ring. They break the beta-lactam ring. So we called these enzymes secreted by the bacteria beta-lactamases. These beta-lactamases, if they are some of them destroy more penicillins, we call them penicillinases. Penicillinases. And there are some beta-lactamases which preferably more destroy cephalosporins. We call them cephalosporinases. And then there are some beta-lactamases which love to destroy, it's very sad now, they love to destroy the carbapenems. We call them carbapenemases, right? Now, come back. As I mentioned, in 1940s, when we started using natural penicillins, right, we were very happy. But unfortunately, bacteria came to a resistant type of bacteria developed, and those resistant bacteria started secreting beta-lactamases, and now natural penicillins were destroyed. And we were very sad. Why we were sad? By 1960, many of the bacteria were secreting what? Beta-lactamases and destroying the natural penicillins, and medical community was sad. Then what happened? We made some extended spectrum and some semi-synthetic antibiotics, right? One of them was methicillin. And similar some other drugs were also made which were added to the group of methicillin, like neficillin and oxacillin, right? In 1960s, methicillin was available, and that was a great happy news for the doctors. You know what? They found in the laboratory that those bacteria which were producing beta-lactamases and which were resistant to natural penicillins because they had the capability to destroy the beta-lactam rings, those bacteria were killed by methicillin. Those bacteria were killed by methicillin. Why? Because methicillin structure was altered in such a way that bacterial beta-lactam could not destroy beta-lactam ring. Now we were clever. What clever thing we did as a doctors or scientists, that we modified the penicillins in such a way that the beta-lactam rings were protected from the attack of beta-lactamases. And this group which came in the market, now it is not available, methicillin now, commercially it is not available, but at that time, methicillin, then neficillin, then oxacillin, and others. Doctors discovered that these antibiotics were able to kill those staphylococci which were producing beta-lactamases. And the reason was that methicillin chemical structure protects the beta-lactam ring in such a way that beta-lactamases cannot destroy. And that was a celebratory time in medical community. Medical community was celebrating that, wow, we have developed a type of penicillin which cannot be destroyed by beta-lactamases. So we said we are having beta-lactamase resistant penicillin. What we were having? We were having beta-lactamase resistant penicillin. But this happiness was very short-lived. We started using these beta-lactamase resistant penicillins or methicillin in 1960. And as early as 1961, some strains of Staphylococcus aureus were discovered which were even resistant to methicillin. Oh my God, that was a very sad time for medical community because now bacteria has done something, has altered itself in such a way that our penicillins, which were beta-lactamase resistant penicillins, even they were unable to kill the bacteria. What was the reason? And soon we found the reason. The reason was that some Staphylococcus aureus from other microbes, they acquired a gene called Mech A or Mech C. Once they acquired this gene, then such bacteria, not only they were producing penicillin binding protein 1, 2, 3, and 4, but due to presence of Mech A gene, now these bacteria were able to produce a very special, very dangerous, very altered protein, which was called penicillin binding protein type 2A. Right? So what happened in 1961? That a strain of Staphylococcus aureus was discovered, which was having a Mech A gene and which was expressing altered penicillin binding protein, which was called penicillin binding protein 2A. And problem was that methicillin or neficillin or oxacillin or similar drugs were not able to bind with the penicillin binding protein 2A. Our methicillin was unable to bind with this altered penicillin binding protein 2A. It means that if we have given methicillin to these patients or oxacillin or neficillin, this will bind with penicillin binding protein 1, 2, 3, 4 and make them dysfunction. But because Mech A gene is there and penicillin binding protein 2A is on the surface and our methicillins or neficillin or oxacillin are not able to bind with these altered new penicillin binding proteins. So these penicillin binding protein 2A kept on doing their normal function. What was their normal function? Transpeptidation. So even though other penicillin binding protein 1, 2, 3, 4 failed, but this altered penicillin binding protein 2A, which was produced due to Mech A gene, this was not having very little affinity or very low affinity to bind with the penicillins, including the methicillin and related drugs. So they remained functional. They keep on doing their transpeptidation function. It means that penicillin binding protein 2A kept on cross-linking the peptidoglycan chains which were being secreted by the bacteria, and bacteria was still able to synthesize a strong, robust cell wall, right? And now we will say this bacteria is not killed by methicillin, right? So we named this bacteria at that time Methicillin-Resistant Staphylococcus aureus. So methicillin-resistant Staphylococcus aureus is a classical example, where target of the antibiotic is altered. Target of the antibiotic is altered in such a way that it will not bind the antibiotic but still keep on doing its normal biological function and cross-linking the proteins. Right? So that was a big problem, and that was the first problem. So all those bacteria, initially we thought in those days that those bacteria which have Mech A gene and they have altered, they are also having, of course, genotypically they are having Mech A gene, and phenotypically they are expressing a penicillin binding protein 2A. They are resistant to methicillin. So, okay, at that time we call them methicillin-resistant Staphylococcus aureus. But very soon we were in for a very big horror. The horror story was this, that very soon doctors discovered such bacteria will not bind any of the beta-lactam antibiotics. Wow, that was the master stroke by the bacteria. Previously, what bacteria was doing, we were throwing the penicillin, they were throwing the penicillinases and destroying our penicillins. We were throwing beta-lactam antibiotic, bacteria were throwing beta-lactamases. Now bacteria altered the target of beta-lactam antibiotics, but transpeptidation was going on. And now these were the bacteria which were very dangerous. It was very difficult to kill them, right? Because initially we thought they are resistant to, yes, methicillin, right? Here I want to mention something. These days when we are checking in laboratory methicillin-resistant Staphylococcus aureus, we check the colonies for the resistant to oxacillin, because methicillin is presently not available commercially. So due to honoring its history, we still call this nasty bacteria methicillin-resistant Staphylococcus aureus. But if someone asks me what should be the name of this nasty bacteria, MRSA, I will say it's a little long name, but it shows how nasty it is. The long name should be almost all beta-lactam antibiotic resistant Staphylococcus aureus. Right? Now, question is that in the beginning, doctors were saying, for example, in 70s or 80s or 90s, doctors were saying that these bacteria are, even though methicillin-resistant Staphylococcus aureus called, but good doctor understood the real concept that methicillin-resistant Staphylococcus aureus were resistant to every beta-lactam antibiotic. But these days, some very good doctors, and you will come across such very good doctors, they will say MRSA are resistant to almost all beta-lactam antibiotics. Almost all, not all, because there are few exceptions. Recently discovered 5th generation cephalosporins, I will mention their name, it's worth it, recently discovered, and now they are being used also. 5th generation cephalosporins like ceftaroline, 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 or more recently discovered ceftobiprole, 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 biprole. So now we have two exceptions at least. There are two 5th generation cephalosporins which are able to bind with these altered penicillin binding proteins. It means these two 5th generation cephalosporins are able to bind and inhibit the function of penicillin binding protein 2A, which are encoded by Mech A gene or Mech C gene. So we can kill MRSA by ceftaroline or ceftobiprole. And there are some other drugs. The management of MRSA I will discuss in other lectures. Now, question is that we have already discussed that any Staphylococcus aureus which has Mech A gene or Mech C gene, it is MRSA. And the one of the fast way to diagnose is if you want to check, do the PCR test on the Staphylococcus aureus for presence or absence of Mech A gene. If Mech A gene is present, this is MRSA, number one. Number two, we learned that Mech A gene produces an altered penicillin binding protein 2A, right? And this altered penicillin binding protein 2A, it can be phenotypically, it can be identified by latex agglutination test, right? So these two tests are important, that Mech gene discovery or its presence or absence is done by the PCR test, or penicillin binding protein 2A, its presence or absence is determined by latex agglutination test, right? If Mech A gene is present, or you have confirmed the presence of penicillin binding protein 2A, it means you are dealing with MRSA, right? So this is the very basic concept of MRSA. But what makes it superbug? The first requirement to make a staphylococcus superbug is that it must have Mech A gene. It must express penicillin binding protein 2A. It must be not only methicillin resistant, it must be resistant to almost all beta-lactams except two beautiful exceptions, that is ceftaroline and ceftobiprole, right? This is what we have learned up to now. Now, one more thing which I would love to mention is that initially these MRSA bacteria were discovered in the hospital environment where vulnerable patients were there, right? But later on, they were discovered in the community also. So basically, on clinical basis, MRSA are divided into hospital-acquired MRSA, and second type is community-acquired MRSA, and now we have livestock acquired MRSA. Now, there's a huge difference in pathogenicity, virulence, and management of hospital-acquired MRSA and community-acquired MRSA. Of course, I will discuss that in detail in next lecture, right? Now, you have any question? Yes, one more thing, that someone was asking me that where exactly in the chromosome of Staphylococcus aureus of MRSA, where exactly Mech A gene is present. Let me tell you something about the Mech A gene. Mech A gene, it is basically acquired, rather it is a part of a bigger DNA length, and that DNA length is called chromosome island. So we can say that, as you know, bacterial chromosome is a circular chromosome. Within this, if you are having a circular chromosome of MRSA, then in MRSA chromosome there will be special chromosomal segment integrated in the circular chromosome. This special segment of DNA or piece of chromosome, right? This is called chromosomal island. And in case of, now listen, in case of Staphylococcus aureus, then within this island, this chromosomal island, there is staphylococcal cassette chromosome. Again, let me repeat it. I know this is confusing. It was confusing for me and also. Let's imagine that you are on an island right? You go to the, yes, where you go? Yeah, somehow you have a huge big Staphylococcus aureus MRSA. You enter into that bacteria and you go on the chromosome, and you are moving on the circular chromosome. There you find one chromosomal length, and it is written that this is chromosomal island. Within this island, there is a group of genes. Within this chromosomal island, there are groups of genes which are called staphylococcal cassette. Cassette, CASSETE. Chromosomal staphylococcal cassette chromosome. So you have number one, you have what? Chromosomal island. In the island, you have group of genes which are called staphylococcal cassette chromosome. Within the staphylococcal cassette chromosomal segment, you have Mech A gene, and along with the Mech A gene, you are also having their regulatory genes. Mech A gene has some regulatory genes like Mech I and Mech R1. You don't need to remember, you just remember, but don't forget Mech A. Mech A, presence of Mech A is what makes a Staphylococcus aureus MRSA. But what I'm saying, Mech A has two associated, there can be Mech A or slight variant which is called Mech C, C for cat, A for apple, of course. So Mech A or Mech C, they are having some regulatory genes with it, which are called Mech I or Mech R1. These genes are also having some other genes related with virulence or some other genes which produce resistance against those antibiotics which are not beta-lactam. And all these collection is called, in the staphylococcus, it is called staphylococcal cassette chromosome. So, and staphylococcal cassette chromosome is part of genomic island. And this genomic island, now what is genomic island? Normally, when bacteria divide, bacterial personal chromosome, when one bacteria become two, it vertically distributes into two daughter cells. But this genomic island or this special type of DNA, which we are calling genomic island, which is having staphylococcal cassette chromosome, this can multiply and move horizontally. What does it mean by horizontally? That from one bacteria to the other bacteria to the other bacteria which are not its daughter cells. For example, if you are a bacteria and you are having, what is this? Chromosomal island containing, you are having chromosomal island containing staphylococcal cassette chromosome, which is itself having at least Mech A or Mech C gene, right? And some other genes also. Now, if you are having this, this can move horizontally. It is not going to your offsprings. You can gift it to others. You can gift it to your friends. How this gift can go? One way. When a bacteria die, this chromosomal segment, when a bacteria is disintegrated and dies, bacterial chromosome also disintegrate. This island might be taken up by some nearby recipient bacteria, and those bacteria also become resistant to methicillin, or those bacteria also become resistant to most of the beta-lactam antibiotics. Is it clear? This horizontal movement is called transference. It is called transference. Then another horizontal mechanism. One bacteria which is having Mech gene, it makes some copies of Mech gene, and this bacteria make a long pilus. It makes a long pilus which connect to a small hole in other bacteria, right? And through the pilus, donor bacteria donates the Mech A gene or the full island to the other bacteria, and other bacteria also becomes resistant to the methicillin, or more accurately, resistant to most of the all beta-lactam rings antibiotics. This mechanism of transfer of Mech A gene, or the mechanism of transfer of resistance to one bacteria to the other bacteria via the pilus, this is called conjugation. This is called conjugation. So these bacteria, either they die and other bacteria pick up their resistant genomic material, we call transference. That new recipient bacteria has undergone transference and now it is resistant. Or one bacteria is resistant, having this genomic island and Mech gene. It makes copy of Mech gene, expresses a pilus which connects with other bacteria and passes the Mech A gene to them, or whole island, genomic island there, or chromosome island there, or we can call it staphylococcal cassette chromosome there. And new bacteria which will acquire this genomic material, that will also become resistant to beta-lactam antibiotics, right? If that is Staphylococcus aureus, then we'll say that bacteria which was previously methicillin-sensitive Staphylococcus aureus, after receiving the Mech A gene, this has been converted into Staphylococcus methicillin-resistant Staphylococcus aureus, right? I've told up to now two mechanisms. One way was transference. I was telling how the chromosomal island move. These are mobile genetic genomic material. How they move horizontally. One way is transference. Other is conjugation. Third is transduction. It has nothing to do with the duct, but it is called transduction. In mechanism of transduction, what happened? There is a Staphylococcus aureus, which is actually MRSA, having Mech gene happily sitting, and some viruses land on that bacteria. And those viruses inject their DNA into bacteria. Such viruses are called phage viruses. What are they called? Phage, phage viruses. So sometimes some some phage viruses, PHAGE, phage viruses, which are able to connect with the bacteria and inject their DNA into bacteria. Then that bacterial DNA replication machinery will multiply many copies of viral genome and also make viral capsids, and then new genome of virus and capsid will repack, and bacteria will release many new virion, many new viruses, right? Sometimes what happens, that if some phage virus has landed on MRSA and injects its DNA into MRSA while its DNA is multiplying, viral DNA is multiplying, and new capsids are being made, in the end, ideally what should happen? That viral genome and viral capsid should reassemble and go away. But sometimes our mobile genomic material, our chromosomal island, Staphylococcus, which is called Staphylococcus cassette chromosome, which has Mech A or Mech C gene with some regulatory components, and even some other genes related with the virulence or with the resistance against other than the beta-lactam antibiotics. This genomic material inadvertently, unfortunately, by mistake, is packed into viral capsid. So few viruses come out of this or bud out of this bacteria which are not loaded with viral genome, they are loaded with Mech gene, and then they go to some other Staphylococcus aureus, which is methicillin-sensitive Staphylococcus aureus, and this viral capsid, which was loaded with Mech A gene and associated genes, if it inject its genome into methicillin-sensitive Staphylococcus aureus, then this methicillin-sensitive Staphylococcus aureus will acquire what? Mech A gene via the process of transduction, via the bacteriophage, right? And now it will be able to express penicillin binding protein 2A. And now this bacteria, which was previously methicillin sensitive Staphylococcus aureus, now it has converted into methicillin resistant Staphylococcus aureus. And now it's going to be our headache either in the hospital environment or in community acquired, right? So next lecture I will be coming, and especially, do you have any question? Otherwise, I will tell what about the next lecture.
What is the difference between gram-positive and gram-negative?
You want to know about the differences between gram-positive and gram-negative bacterial cell wall?
Yes.
Okay. I will tell you those differences. They are very, I will just few differences. You know, gram-positive bacteria, they have more thick cell wall. Usually, gram-positive bacteria have one cell membrane, and outside that membrane, they have very thick cell wall made of peptidoglycan cross-linking, and this is usually 50 to 80 layers thick, right? But in gram-negative, there is inner membrane, and then there is outer membrane, and in between inner and outer membrane, there is a space called periplasm, and within this space, peptidoglycan, 2 or 3 layers, very small layer is present over there. So if someone asks that gram-positive bacteria cell wall is thick or gram-negative bacteria cell wall is thick, of course gram-positive bacteria cell wall is thick. Small gram-negative bacteria cell wall is thin, but outer to the thin cell wall, it has additional cell membrane which has a lot of polysaccharides, which are also called endotoxins, right? So the special outer membrane, which is present only in the gram-negative bacteria, this outer membrane have some very important structures. They are having polysaccharides, endotoxins. Polysaccharides should be called full name lipopolysaccharides, which are also called endotoxins. Also, they have pores, which are called porins, through which nutrition and drugs can go in and out, right? And if you come to gram-positive bacteria, they do not have outer membrane. So, of course, gram-positive bacteria do not have porins, which is a feature of outer membrane of gram-negative bacteria. Gram-positive bacteria do not have lipopolysaccharides, because that is a feature of gram-negative outer membrane. But gram-positive bacteria have special type of molecules. We call it teichoic acid. Teichoic acid. Now, some teichoic acids are called wall teichoic acid. What are wall teichoic acids? These are special antigens which are small threads like antigens, which are present only in peptidoglycan layer. We call them cell wall teichoic acid. But some teichoic acid are present in cell wall, but they go very deep and they are connected with the lipids of membrane, inner membrane, in the Staphylococcus aureus. These are called lipoteichoic acid. So gram-positive bacteria have in their cell wall teichoic acids, and from the cell membrane throughout the cell wall, they have threads like antigens which are called lipoteichoic acid. And gram-negative bacteria, outer membrane, they have lipopolysaccharides or endotoxins and porins and many other things. We will not talk now. But your answer here, I want to make it very clear that gram-positive bacteria have very thick peptidoglycan layer. They have inner membrane but no outer membrane. Gram-negative bacteria have inner membrane, also have outer membrane. In between the two membrane, there is a sandwich thin layer of peptidoglycans, and that space is called periplasmic space, where they have some digestive enzymes, antibiotics also come there, and many other functions. We'll talk sometime later. Right now, what I want to say, that next time I will continue with the lecture on MRSA, right? And we will especially focus on the differences between hospital acquired and community acquired MRSA, their clinical features, the mechanism of acquisition, their molecular differences, their resistance differences, their virulence differences, right? And how to treat those bacteria. But here I would like to repeat one thing. Why we call MRSA a superbug? Simplest explanation and absolute explanation is that any Staphylococcus aureus which is having Mech A gene or similar Mech C gene and expresses penicillin binding 2A antigen, which lead to resistance to most of the beta-lactam antibiotic, that is MRSA. But making MRSA such a hardy bacteria, there are a few more factors. Over the decades, MRSA bacteria has not only has first step, that is Mech A gene or C gene, it has also acquired resistance genes against those antibiotics which are not beta-lactam antibiotics, especially hospital-acquired strains. And over the decades, they have also acquired genes for very special type of very dangerous toxins. One of the toxin is penton valentine leucocidine. This is a toxin. I will discuss that in next lecture. That some of the strains of MRSA, especially which are acquired, community acquired, they have acquired penton valentine leucocidine virulence factors. That also makes these bacteria more difficult to manage. So it is not only about that these bacteria have Mech gene and 2A penicillin binding protein and they are resistant to most of the beta-lactam antibiotics. These days, some of these superbugs are resistant to many antibiotics, and I'm going to teach you in the next lecture, right? And many of these strains have acquired new type of, what is this? Virulence factors like penton valentine leucocidines, and some of these bacteria have developed more mechanism to evade our immune system. They develop biofilms of polysaccharide and hide inside them where our complement factors and our antibodies and other immune component cannot go. So having Mech A gene and penicillin binding 2A protein is an absolute requirement to name a bacteria MRSA. But actually these bacteria have acquired many other characteristics which we will discuss in the next lecture, which make it so difficult to manage these bacteria. So let's have a break now. We'll continue later. Thank you very much.